Okay, the review on the protein isoaspartyl methyltransferases was part of a larger volume on protein methyltransferases. There are many, many methyltransferases, all of which transfer methyl groups to proteins. And in most cases, these have some kind of a regulatory role. Now, my enzyme, the protein isoaspartyl methyltransferases, which I'll call PIMT, are different in that they don't modify what you'd call a normal protein. They modify a protein that an amino acid that has been generated as part of the aging process that proteins undergo in cells. Just like anything, proteins are subject to spontaneous chemical breakdown. And one of the things that happens is there's an unusual amino acid produced. It's called isoaspartic acid. When that's generated in a protein, what that does is it disrupts the shape of the protein. Now, the shape of the protein is critical for its function. Think about a lock and a key. If the two have to be complementary in order to work. So if you change the shape of either the lock or the key, things aren't going to work properly. And that's exactly what happens when you get an isoaspartic acid into a protein sequence. So what this enzyme does is it transfers a methyl group to these damaged spots in polypeptide chains or proteins. And what that serves to do is kind of be a signal for a repair process to be initiated, that there's following the methylation event, there is a rearrangement that takes place that can then convert the polypeptide backbone, and polypeptide is just another name for a protein, back to the normal shape that the protein should have, so thereby restoring function. I kind of think of these as the sort of unsung heroes, these enzymes. It was kind of a serendipitous event that they were even discovered because they're so good at their job. They're very good at repairing the damage that appears in cells, and as a consequence, you don't see it accumulating, the damage accumulating in most cases. And so it really took the right kind of a cellular system in order to even understand the function of the enzymes. Now, we think they're important for aging because, as I said, the site itself is generated during the aging process. And if you look at very old proteins in cells, what you can see is over time they actually do start to accumulate some of isoaspartic acid that the methyltransferase or PIMT is not able to get to, so it's unable to fully repair all of them. And I'll give you an example. You can think about your lens of your eye. Okay, in the lens of your eye, you have proteins that are as old as you are. Okay, now that's pretty old for a protein. And if we look in the very center of the lens into the nucleus, we can see some of these damaged residues that have appeared in the protein. Other proteins that get this kind of damage are inaccessible to the methyltransferase normally in the body. They might be extracellular proteins. Think about collagen. Collagen is your most major protein. Your bones are made of collagen. Lots of your skin has a lot of collagen in it. This is normally outside cells, and so the methyltransferase can't modify it. So in these cases, we start to see some of the isoaspartic acid appearing. But inside cells where the methyltransferase can get to proteins, we don't see this kind of damage largely appearing. And it's probably just because of the efficiency of the enzyme itself. Probably all proteins are subject to the kind of modifications that this isoaspartic acid formation. If you take any protein and you just kind of put it on the shelf, for example, over time, you'll start to see isoaspartic acid appearing. The biotechnology industry is finding lots of this appearing because they make all these protein products, that they're going to use as drugs, and they put them on the shelf, and then they need to sell them. And what they'll find is over time, they start to get some of this isoaspartic acid appearing in the proteins. Now, if that was in a situation where methyltransferase had access to the protein, like inside the body, those isoaspartic acids wouldn't normally be accumulating because it's a damage that the cell needs to take account of and to repair somehow. So that's why we think it's so important. All proteins are subject to this kind of modification. The methyltransferase is real good at its job. You don't see the damage accumulating. We can look phylogenetically and see where you see the methyltransferase. And when you do that, we find it almost in all organisms that live on Earth. There are some that don't have it, but we classify it as an ancient enzyme. We find it in the bacteria. We find it in some of these microorganisms that live in extreme environments, which you call archaea. Because they're very old in terms of their origin, and in all of the multicellular eukaryotes, you find the methyltransferase. So in us, in plants, everywhere we find it. We're still trying to figure out, truly its function outside of the chemistry that we've already established, but we really want to now figure out what the physiology of the enzyme, what the physiological importance, I would say, of the enzyme. Yeah, we study these methyltransferases in a lot of different ways, and that's really kind of a reflection of how you do biology these days. Everything is becoming much more interdisciplinary. We do a little bit of chemistry, we do a little bit of biology, and so the original studies were primarily biochemistry. The enzyme was purified. We and others have crystallized some of the enzymes to get an idea of what their actual three-dimensional configuration is. Remember that lock and key that I was talking about earlier? You want to understand what the real conformation of the protein is. Some of the biochemistry involves identifying what proteins the methyltransferase prefers to modify in cells. Some proteins are more susceptible to the kind of damage that generates this isoaspartic acid. In particular, flexible areas of proteins, and some proteins have certain regions that are always kind of flopping around a little bit. Proteins in general move, but some regions of proteins are much more flexible. And these regions are the ones that are most susceptible to the formation of isoaspartic acid. And so that's the biochemical view. Other things that we do from the biological perspective is we try to look at what happens when you disrupt the function of this protein. So we're in the era of transgenic organisms, and so we now have a number of genetic models where the protein has been inactivated. These include a lot of the model organisms. So it's been inactivated in bacteria. We've done it in yeast. It's been done in the fruit fly. It's been done in Cynorhabditis elegans, which is a little nematode, which is a nice model. It's also been done in the mouse. And those studies are giving us some really interesting data. They're telling us, actually, that the enzyme is not completely essential for life. You can live without the methyltransferase. Now, so it might be kind of like your 50-year flood enzyme. Cells are susceptible to lots of different kinds of stresses. When we do things in the laboratory, we really keep organisms under pretty rich conditions. It has nothing similar to what they encounter out in the wild. And so, the question is, what's the real physiological stress that the organism really encounters? The other thing we have to think about is what other genes work together with it. Is this methyltransferase part of a gene network? And so we need to have some genetic models where we can start to look at interactions the genes. We talk about this as sort of your genetic background. The same gene doesn't always operate the same in two organisms of the same species, depending on what other genes that these two individuals might have. We have a couple models that we're using in my lab to try to address this. We use the fruit fly as one model. So here we have a nice multicellular organism. You might not think there's much similarity between a fruit fly and us, but we know that actually the basic way in which the body plan established is very similar from fruit flies to man. It's been a very strongly conserved body plan patterning mechanism that is in a lot of bilateral organisms. In the fruit fly, what we're able to do actually is we've overexpressed it. We've made fruit flies that have too much of it. And they live about 50% longer. My father always asked me why we want fruit flies to live 50% longer, but it's because we think that they make a nice model for us. So it's not essential. We also have a knockout in the fruit fly, but it clearly can extend lifespan under certain circumstances. So our challenge in the fly is to identify the particular kind of stresses that make conditions where you require the methyltransferase. It's not necessary all the time, but under certain situations we think it probably is important. One of the areas we're getting very interested in right now is the formation of some of these protein aggregates that occur in diseases like Alzheimer's disease, and amyloid proteins we know are very susceptible to isoaspartic acid formation. We're doing a little work with prion proteins in the lab right now, and there's a number of models in the fruit fly, believe it or not, that are models for human diseases that involve aggregates of proteins formed that have abnormal proteins associated with them. The other organism we're using is yeast, and you say, well, yeast, that's a pretty simple organism. So it's a eukaryote, which means it has a nucleus, just like we do, but it's only a single cell. The beauty of the yeast is that we have so many genetic tools available for it. We know what all the genes are. We have lots of methods for putting genes together in new combinations. And so the hope is, with the knockouts that we have in yeast, that we can look at interactions between families of genes and use that as a tool to try to get at the kind of stresses that make this methyltransferase so important. Because, why keep this enzyme all through evolution unless, it's filling some kind of very fundamental function. So we hope with these kind of genetic models that we'll come to some understanding of what that's all about. But I think the biggest challenge right now is, we're in what we call the post-genomic area in biology, where we now have all these genomes that are fully sequenced and what have you. So we have a huge amount of information to work with, and we have a huge number of genetic variants that we can start to play with. So I think the challenge right now is really on this physiological area to really identify what the function of it is in a combination of different genetic backgrounds. It really isn't proven. We know it's not essential, but there probably are. We have to identify the conditions under which it really does become an essential gene. And to do this, we can take advantage of some of these tools that are available because of the various genome projects that have been done. They've now got everything now in biology is done kind of on a high throughput scale. So, the ability of having a mutant, you can take your mutant now and you can say, well, okay, if I disrupt the function of my gene, which in our PIMT in our case, how does that affect the expression of all the other thousands of genes in the organism? And there are now microarray methods and other kind of high throughput techniques where you can actually begin to kind of answer these questions. And so I think that's where we're going next, and it's going to involve a lot of complexity. I think what we're finding with these microarray type experiments and what have you is you generate a huge amount of data and then you have to mine through it, so it's going to take us a little while. So it's going to be a challenge, but it is going to be defining these networks that are the methyltransferase is participating in. Okay. It's true that there really are small numbers of women scientists here at Boston College. It's not unlike departments in much of the country. Biology has more women faculty than do the physical sciences. Chemistry and physics, for example, I think there's only a single woman on each of those faculties. And that's pretty much what we see nationwide. For whatever reason, historically more women have gone into biology. Times are changing. I look at our students and I see that over 50% of our students now are women. And I think the culture has changed a lot, and the motivations that women have for choosing careers has changed quite a bit since I became a scientist. I think any of us look back in our career and you say you had a whole lot of lucky accidents, and to try to define what brought you to where you are is somewhat difficult. Now, it's interesting. My dad was a chemist, and my mom, was a housewife, and many of the women were in the 50s and the 60s. And the statistics say that a lot of women scientists actually had fathers who were scientists. So I don't know. So the modeling may start at home, and you got your father as a model, you got your mother as a model, and you do little bits of both of them, I think. And then I think another thing that was important. So I decided I wanted to do science. And when I went to college, it was like, well, why don't you do biology? That was the suggestion that they gave me. So I said, oh, that sounds good. I really wasn't fixated on one kind of science versus another. And then I really found my niche in biology. Another really important feature in women going on in science, anyone going on in science, I think, is doing research as an undergraduate student. I was asked to join in the honors program when I was at Purdue, and a part of that was to do undergraduate research. It's something that I might never have thought of on my own, but it's really, I think, an important part of a career. It can be a really motivating feature for, or motivating factor for young people. So my advice to women would be to when they open themselves up to a range of experiences, to seek out opportunities to do research, to seek out other women who are in the sciences as well. I do think that there are sort of unique challenges to being a woman in science. The biggest one, of course, is balancing work and family. I have now 2 grown children, but it was a juggling act in those younger years. I think people have, the culture is easier now, in terms of daycare possibilities. It's more socially acceptable for women to work than it was when I first started. But that balance of family and research is always a very tough one. I think that one that's never going to go away. I was really glad to find out about e-scholarship here at Boston College, because the book, The Enzymes, is a well-regarded series, and many libraries, it's been around for quite a few years. We were volume 24 in the series. Just because of both budgetary and space considerations, a lot of libraries now don't have complete subscriptions to it, so people were not going to find it on their library shelf. Another major problem that we had was that the articles in the enzymes, even though the series is well known, are not accessed in most of the bibliographic databases like PubMed, for example. And so I was concerned that the chapter would be falling into oblivion, and only a very specialized group of people would really even know that it existed. And I had written it really to be a very comprehensive review of the field, so that was a great disappointment. And Sally Wyman, our librarian, suggested to me that I might find out from the publisher if they would allow me to put it into e-scholarship, which is our data, I guess you'd call it our database here at Boston College. And so I contacted the publisher, which was Elsevier, and they were quite willing to let me put a preprint version of the article up on e-scholarship. So I did that. It was very easy because, of course, we all now prepare our own manuscripts, so we always have these sitting on our desktop anyways. Submitted it, and like a day later, it was out there and available to the public. It's only been out there for a month, and already I've had something like 14 downloads happen, and I've had some regular reprint requests come through. So I think, for a month it's really been quite successful, and I have a link now to it from my own personal web pages as well. So I'm very grateful for the opportunity to have it out there. Most scientists these days don't go to the library a whole lot. They go physically to the library. They like to go electronically to the library, so being able to download those PDFs is really the way that we get most of our information. So I'm actually very fortunate that the article was published by Elsevier, because they do, because when you write a manuscript like that and submit it for a book chapter, you sign away your copyright rights. And so in many of the publishers that don't allow you to then make these manuscripts available to the public, but Elsevier has been one of the leaders among the publishing companies on allowing authors to put preprint copies of their manuscripts on their institutional repositories. And so this is really the critical difference, and I think it does a lot really to further science. I put a link to the Elsevier webpage advertising the book on my own e-scholarship submission, and that's good for Elsevier too, because it actually directs people to their site, because this is really part of a much larger volume that has information on lots of the other methyltransferases. And so people who are interested in my enzyme are probably interested in some of these others as well. So I think it's kind of a win-win situation for both the publisher and also for me, and also for the scientific community at large.